Alright, in this video we are talking about the nucleus, which is an organelle, but it is a special organelle, so it gets its own unique little section. So we're going to talk a little bit about the nuclear membrane, the membrane enclosing the nucleus. We're going to talk about what's inside of the nucleus and how DNA is organized within the nucleus. So the nucleus is the largest organelle in a human cell. It is where all of the DNA is stored, and DNA stands for deoxyribonucleic acid, which is fancy word for all of our genetic information. That is how we store all of our genes that control how we function, when we do things, what we look like, and tons of other stuff. Some cells, like skeletal muscle fibers, are going to have multiple nuclei in them, so more than one nucleus. They are called multinucleated. And then some cells are actually going to have no nucleus, and these are red blood cells. They are anucleated. They are actually going to start off early on in their life cycle with a nucleus. Once they fully finish developing and are ready to start carrying gases around, oxygen and carbon dioxide, they are actually going to eject their nucleus, and they then become just bags of hemoglobin because that nucleus takes up a lot of space and they need as much space as possible to transport oxygen and carbon dioxide around the body. So they ultimately do not have need for a nucleus. So the nucleus is surrounded by a double lipid bilayer called the nuclear envelope, or envelope, depending on how you want to say that. Between these two layers is nuclear pores. So these are openings in that lipid bilayer that are going to allow movement of different materials in and out of the nucleus. There's not a lot of things that are going to go in and outside of the nucleus, but proteins, RNA, and certain solutes, certain ions, are going to need to go in and out of the nucleus. So proteins, really essential for the function of the human body in so many different ways. We do need proteins to be able to get in and out of the nucleus. RNA also needs to get in and out of the nucleus. That's kind of the in-between our genetic information, those genes, and the stuff that those genes are controlling. So they are going to help create some of the proteins that we're making that are controlling our appearance when we produce certain hormones, when we produce certain enzymes. Proteins are also going to make up some of our hormones and our enzymes. Inside of that nuclear envelope is the DNA. DNA is organized into chromosomes. Chromosomes do only exist during cellular replication. We'll talk more about that in another video. Chromosomes, though, are the condensed form of something called chromatin. Alright, so chromatin is how DNA normally exists when the cell is not going through mitosis. Alright, so this is a graphical representation of the nucleus. So you can see we've got our nuclear envelope here, we've got two layers to that, and we've got our rough ER out here that is joined with that second outer layer of the nuclear membrane. And then you can see embedded in those layers of the membrane are these nuclear pores so that different substances can get in and out of the cell. Floating around inside is the chromatin. So all of this is our genetic information. So it's kind of loose and uncoiled in its normal state outside of cellular replication. And then far deep down inside is the nucleolus, which is where a lot of that information, that genetic information, is stored. So DNA structure, like I said a couple slides ago, exists primarily as chromatin and then sometimes as chromosomes. So DNA is made up of all of these nucleic acids, adenosine and guanine, cytosine, and why am I drawing a blank on what T stands for right now? It'll come to me in a minute. A, T, G's, and C's are the abbreviations. These are also called base pairs. And so A and T will always be linked together, and then C and G will always be linked together. So T and G would never link together, A and C would never link together. There are specific pairings for these based on the molecule structure of each of these bases or nucleic acids. And so we have this double helix, so we have 2 rows of AT, Gs, and Cs that are going to be bound to each other with a connection between hydrogens, forming this kind of 2 layers of ribbons that are going to form into a spiral structure that's called our DNA helix. And we are also going to add in histones that are going to be kind of helping to bind DNA together and keep it all packaged up. So chromatin is a very loose structure of this DNA molecule because it takes a lot of energy to actually maintain this chromosome structure. And so if we're not needing that chromosome structure to do cellular replication, then we don't want to be expending a bunch of energy trying to keep it in that particular structure, so it's going to relax a little bit and stay in this chromatin structure. That's pretty much it for the nucleus. Nice short video here. In our next video, we will talk about cell growth and division, so we're going to do a little bit deeper dive into DNA and chromosomes and chromatin and when each of those structures are going to exist.